All right, let's dive right into this explainer. Today, we're taking a pretty wild journey. We're going to start way down in the microscopic, physical reality of our cells and zoom all the way out to the macroscopic math of how traits actually get passed down. We are decoding the literal mechanics of inheritance, and honestly, it's fascinating stuff.
Here's our roadmap for today. First, the cellular coin flip. Second, the math of inheritance. Third, real-world supercrops. And finally, number 4, when the rules break.
So, let's kick things off with part 1, the cellular coin flip, inside metaphase I.
To really wrap our heads around genetics, we've got to look at the actual cellular machinery at work deep inside the cell during a very specific phase of division called metaphase I. Notice how the spindle fibers are attaching to the kinetochores on the chromosomes? They're basically gearing up to pull each chromosome pair to opposite poles of the cell. But here's the absolutely critical detail. The specific pole that each maternal or paternal chromosome attaches to, it's completely random. It is, quite literally, a biological coin flip.
And that brilliantly illustrates the sheer power of this randomness. Because human cells have 23 chromosome pairs, the cellular coin flip multiplies across all 23 pairs. 2 to the power of 23 generates, get this, over 8 million unique possible gamete combinations. Every single gamete a human produces is drawn from this incredibly massive pool of random possibilities. It's just mind-blowing.
This random sorting mechanism? That is the exact physical basis for the law of independent assortment. Basically, it's the principle that the segregation of one chromosome pair happens entirely independently of the segregation of any other pair. It ensures that maternal and paternal genes mix independently, which guarantees a massive shuffling of the genetic deck every single generation.
Moving right along to section 2, the math of inheritance, from cells to Punnett squares.
Now we're zooming out from the physical cell to look at the mathematical combinations that predictably emerge in the offspring. Let's trace a dihybrid cross, which is really just a fancy way of saying we're looking at the simultaneous inheritance of 2 totally distinct traits, like seed shape and seed color. Step 1, we cross pure, true breeding parents. Step 2, that gives us a uniform, heterozygous F1 generation. And finally, step 3, those F1 offspring self-fertilize, which lets us analyze all the highly varied combinations in the F2 offspring.
So because the alleles assort independently, like we just talked about, they produce 4 equally likely gamete types. We're talking a 25% probability for each. When you cross these equally likely gametes, they combine into this massive matrix that yields a very precise 9:3 to 3:1 phenotypic ratio. That's 9 round yellow, 3 round green, 3 wrinkled yellow, and 1 wrinkled green. Seeing this precise 9:3 to 3:1 ratio is actually the classic signature proof that 2 genes are assorting independently. To make this math even more intuitive, this complex 16-square ratio is actually just 2 independent probability coin flips multiplied together. The product rule tells us that a 3:1 ratio for seed shape multiplied by a 3:1 ratio for seed color perfectly calculates out to 9:3 to 3:1. It's beautiful math, proving that these genetic traits are absolutely not influencing one another.
But hey, don't fall into a super common conceptual trap here. Just because there are 16 combinations mathematically possible definitely doesn't mean they occur equally in nature. While the gametes themselves are indeed produced in equal 25% proportions, random fertilization means heterozygous combinations are mathematically way more likely. They actually occur in a highly varied, unequal pattern that heavily favors mixed traits over pure ones.
All right, on to section 3, real-world supercrops and engineering agriculture.
Let's take all this abstract math and apply it to something incredibly tangible. Plant breeders rely heavily on these exact dihybrid and multihybrid crosses to develop supercrops. By hunting through thousands of F2 generation plants, they're looking for that one perfect, statistically rare genetic combination. For instance, finding a single crop that has independently inherited both high grain yield and extreme drought resistance. It's like finding a needle in a genetic haystack. And modern technology has completely supercharged this. Instead of waiting for a physical plant to fully mature to see its traits, scientists now use marker-assisted selection. By looking directly at the DNA markers, they can identify those favorable, independently assorted genotypes immediately. This has literally shrunk a traditional decade-long breeding process down to just 3 or 4 years. But, here is a critical plot twist to keep us on our toes. Does this neat and tidy mathematical rule of independent assortment actually apply to every single gene in a cell? What do you think?
That brings us to our final section, number 4, when the rules break, covering linkage and exceptions.
So the fundamental divide here all comes down to location. Genes on different chromosomes assort independently, exactly as we've calculated. But genes that are physically located on the exact same chromosome, that's an entirely different story. They are linked together. They actually travel through meiosis as a physical package deal.
When we compare the two mechanical processes, it's pretty stark. Standard independent assortment yields equal gametes, but physical gene linkage completely alters our expected outcomes. Because linked genes are inherited together, unless a rare crossover event happens, this heavily favors parental traits. It results in way fewer recombinant classes than our trusty 9:3 to 3:1 ratio predicts, massively skewing the phenotype data. Basically, the math goes out the window.
Furthermore, sometimes structural cellular failures can entirely override the whole process. Say a spindle fiber just fails to attach during metaphase I. That structural failure forces the chromosomes to align non-randomly, which completely destroys the independent segregation math and results in huge, skewed deviations from our expected ratios.
Before we wrap up today, let's clear up some common diploma traps you might encounter. Remembering the critical difference between statistical expectations over massive samples versus actual reality, and knowing for sure that independent assortment only applies to genes on different chromosomes, well, keeping that straight will save you from some major conceptual misconceptions down the line.
And this brilliantly illustrates why all of this matters so much. This microscopic randomness, it isn't just some mathematical trivia game, it is the very engine of genetic diversity. By constantly shuffling alleles into infinite new combinations, independent assortment provides the raw material necessary for natural selection and evolution across the entire planet. It's the reason life can adapt and thrive. Which leaves us with a pretty fascinating final thought to ponder. If our physical traits are ultimately just an incredibly complex roll of the cellular dice, what hidden, remarkable combinations of traits are waiting to be uncovered in the next generation? Thank you so much for walking through this explainer with me today, and hey, keep questioning the mechanics of the world around you.